Next stage in mitosis is called metaphase. Same thing, we're going to describe what the cell looks like, and then the key words I need you to use when you're explaining this are spindle fibers and the cell equator. So if we had our cell membrane still present, but our nuclear membrane is gone, okay, and remember we had our centrioles here and here, right, on opposite sides of the cell. We also had our chromosomes that were visible, and now what's going to happen is those chromosomes are going to be captured by these things called spindle fibers. And what I want to kind of explain to you is that these spindle fibers are not always present in the cell. They are formed only during metaphase. And what forms them, and here's where we get to the important part that I keep saying it's coming up, is the centrioles. So I like to think of these centrioles as like Spider-Man's web shooters, okay, and they can shoot out these spindle fibers, okay, these little webs that capture our chromosomes. Remember, the chromosomes aren't in the nucleus anymore, right? The nuclear membrane disappeared. So these chromosomes can get captured by, let's see, I think I'll use my thicker marker here. They can get captured by the spindle fibers, and they're kind of sticky, like I said, like spider webs. So here's one set of chromosomes, right? There's two sister chromatids in red, and then I think I did blue. Here's another one captured by the spindle fibers, and I think the last one I did was yellow, again captured by the spindle fibers. And you see, when they're captured by the fibers actually pull them along this imaginary line here that we call the cell equator. So here's our cell equator. Okay, if you imagine like this is the North Pole and the South Pole where the centrioles are, the cell equator goes right across the middle, and that's where those sister chromatids are lined up. Okay, so to go through this again, I'm describing the cell. I'm saying this is the cell membrane. The centrioles are at the North and the South Pole. The spindle fibers get shot out from the centrioles. They stick to the chromosomes, and then they line them up at the cell equator. Right, that's metaphase. Meta, I always think of as middle. Okay, meta is middle. Middle because it's lined up in the middle of the cell. Okay, that is what helps me to remember what metaphase does. Okay, meta is middle. Okay, two more steps. Anaphase. Again, we're going to start with what the cell looks like and then describe the changes that happen using these keywords. So if we start with, again, still our cell membrane present. This will make our marker a little thinner here. Okay. We've got the chromosomes, remember, are lined up in the middle of the cell to begin with. We still have our centromeres, centrioles, and these spindle fibers. Okay, what happens in anaphase, though, is the spindle fibers get shorter. And when they shorten, they actually pull apart the chromosomes. So remember, in the last one we had, our two chromosomes that were red were stuck together in the spindle fibers. Now, the spindle fibers shorten, and those two sister chromatids are getting separated. So all of those chromosomes that originally were attached together are now getting separated apart because those spindle fibers, again, are shortening. Okay, so in anaphase, the spindle fibers shorten, separating the sister chromatids. The place where they separate them is called the centromere. Okay, the centromere is what attaches each chromosome, each sister chromatid, to the other. So like right here in the middle of each one of them was like a little snap, if you can imagine that held the two parts of the sister chromatids together. Well, the spindle fibers unsnap that snap and then pull them apart at that point. So the centromere is like the snap that holds the chromatids together until the spindle fibers pull them apart in anaphase. All right. One more step. Last stage of mitosis we call telophase and also cytokinesis. We kind of lump these two things together. So we're going to start with our cell again. Cell membrane, still there. Let me redraw that. That might be better to start with how that changes first. So the cell membrane is still there, but what happens is the cell membrane starts to kind of pinch in. So you can see this is all one cell membrane, but it's starting to kind of get narrow in the middle there. All right, so that is getting ready to split in half. The centrioles disappear. So I'm just going to write that up here. Remember the centrioles that held the spindle fibers to move those chromosomes around? Now they're going away, along with the spindle fibers. Okay, the nuclear membrane disappears. All right. Let me do a different drawing of a cell membrane so it makes more sense with the last picture I just did. So remember we had our, get these the right way, our chromosomes divided like this. Remember they got split apart. I think I had the blue ones next, but that's okay. They got split apart by the spindle fibers. Okay, so basically we've got two sets of identical chromosomes, right? The top and the bottom. So here's one set of chromosomes, and here's one set of chromosomes. And remember they're identical because way back when they made copies of themselves. So now the cell membrane can split, and if we're going with the picture I just had, it's going to split this way. So I apologize for drawing that other one first. Okay, do you see how that's going to divide it into two identical cells eventually? Eventually when this all splits apart, we'll have two identical cells because they each have a red chromosome, a yellow chromosome, and a blue chromosome. Okay, so the cell membrane is going to pinch in, okay, dividing our two sets of chromosomes in half. As I said, the centrioles disappear because we no longer need those spindle fibers. The nuclear membrane, which remember disappeared way back when in prophase, starts to reform. But now we've got two sets of DNA, right? Two sets of chromosomes, two cells, so we have two new nuclear membranes. So now we've created two cells that are essentially identical to the first one that we started with as far as the DNA goes. The telophase part of this is the actual making of this nuclear membrane. Okay, the cytokinesis part is the splitting of the cytoplasm. All right, so how I describe this is in telophase and cytokinesis, the cell membrane pinches in, dividing it into two cells. The centrioles disappear, and the nuclear membranes reappear. And that's your last step.